What I'd like to do then is give some important considerations about the validation of surrogate endpoints and extend some of the insights just presented by Dr. Jeffrey Siegel. So basically, some key principles: a strong individual level correlation between a structural or inflammatory biomarker with a direct measure of how a patient feels, functions, or survives establishes that that biomarker is a correlate of risk. But as Nikolai presented up front, it's just a necessary but not sufficient condition to be a valid surrogate or replacement endpoint. That is, it doesn't justify that the treatment effect on the biomarker reliably predicts the treatment effect on how an individual feels, functions, or survives. So the validation process then requires an in-depth understanding of the causal pathways of the disease process and the treatment's both intended and unintended mechanisms of action. And critically here, meta-analyses of trials showing that the net effect of the treatment of the biomarker predicts its net effect on how a person feels, functions, or survives. So graphically, in osteoarthritis, we're looking at experimental therapies that can improve how people feel, function, and survive. And as Dr. Kim and others will talk about, this could be a composite endpoint: total knee replacement, severe pain for feels, or severely impaired functioning for functions. And so an experimental therapy may have an effect on feels, functions, survives, mediated through many mechanisms. Unintended effects and intended effects could be imaging biomarkers, joint space narrowing, or other intended effects, inflammatory markers. A given biomarker can be on an individual level correlated with feels, functions, survives, and yet this arrow doesn't exist. It's not a causal pathway where this relationship could exist because the biomarker is correlated with other biomarkers that are on the causal pathway. In this setting, an effect on the biomarker is not going to reliably predict the effect on feels, functions, survives, even though it's an individual correlate. Even if this pathway exists, the relative effects of an intervention on a biomarker may not predict its effect on feels, functions, survives. A classic example of this is in the setting of acellular pertussis vaccines, where the Sweden trial was a 3-arm trial with a diphtheria tetanus control and 2 experimental arms, adding either the Smith-Kline-Beecham or the Aventis Pasteur acellular pertussis components. And at this point in time, FHA and PT were thought to be potential correlates of protection. We talk a lot about correlates of protection nowadays with vaccines for COVID. It was thought that these could be used as registrational endpoints. Interestingly, the Smith-Kline-Beecham vaccine was superior to the Aventis Pasteur on these biomarkers, even though for the clinical endpoint, it had 3 times the number of breakthrough cases of the Aventis Pasteur. And this could be explained in many ways, including the fact that the Aventis Pasteur vaccine not only had the intended effects on FHA-PT, but on other immune responses that resulted from additional antigens in the vaccine, and also by durability of the effects. An effect of an experimental therapy on a biomarker could also overestimate the effect on feels, function, survives, because interventions often also have unintended effects. In this slide, we look at many experimental therapies across an array of different settings, where each of these experimental therapies had its intended effect on the biomarker, and ultimately on feels, function, survives had adverse effects on morbidity and mortality, mediated through the unintended effects they had on increased thrombosis, cardiovascular risks, hepatic risks, or activating the renin-angiotensin system, increasing blood pressure. And this bottom example here of Revatio points out also that the use of biomarkers can result in misleading conclusions when you're extrapolating across populations, for example, from adults to children. So sildenafil in adult pediatric arterial hypertension, in pulmonary arterial hypertension, had evidence that with increasing doses, you had improved hemodynamics and improved clinical benefit at 6-minute walk. But when this was then looked at in pediatric PAH, it's called Revatio, Revatio also showed with increasing doses a good effect on the biomarker, improved hemodynamics, and yet increased mortality. The effect of an experimental intervention on a biomarker can also underestimate the effect that therapy has on feels, function, survives. A classic example of this is in the setting of oncology. In oncology, for decades, we've been studying chemotherapy regimens for their effect on overall survival, often using as a biomarker the tumor burden measures, objective response rate, shrinking tumors, or progression-free survival, delaying the time to increasing tumor burden. In recent years, immuno-oncology agents have had remarkable results when compared to chemotherapy. Yet when comparing these agents using the traditional biomarkers, chemotherapy is better, even though the immuno-oncology agent has improved survival. And that's because the immuno-oncology agent not only improves or has an effect on the short-term tumor burden, but has a very strong effect on long-term tumor burden measures. And in essence, I often say, in this setting, we need more patients CE rather than patients TS. And this could be relevant to our understanding joint preservation endpoints in the OA setting. So a key conclusion here is that it can be treacherous to extrapolate surrogate endpoints across different classes of agents. PFS and objective response were fine as biomarkers for chemotherapy, but not for immuno-oncology setting interventions where long-term tumor burden effects are really critical. So how does one establish that a biomarker would be a valid replacement endpoint for a feels function survives measure? And in essence, we need critical evidence is the net effect of the treatment on the replacement endpoint reliably predicts the net effect of treatment on feels function survives. Classic example, I was serving on the Cardiorenal Advisory Committee in June of 2005, and FDA asked us for antihypertensives, can we now use blood pressure as a biomarker for clinical benefit? And the data that they provided to us was from 43 clinical trials, where each dot here is a clinical trial, a mega patient clinical trial, where for each dot on the x-axis, it was the net benefit of the experimental arm in reducing blood pressure, and on the y-axis, the net benefit on the clinical endpoints. And in essence, why 43 trials to validate blood pressure? And that was because the validation was done separately for each class of agents, for beta blockers, low-dose diuretics, ACE inhibitors, calcium channel blockers, and angiotensin receptor blockers, because it was understood that each of these classes could have a different off-target orange pathway. Interestingly, also, the validation of blood pressure depended on the endpoint. Blood pressure was highly predictive of the effect on stroke, moderately for MI, cardiovascular disease, only somewhat for mortality, and not at all for heart failure. So in essence, the Institute of Medicine had a really important, lengthy report on the evaluation of biomarkers and surrogate endpoints. And one critical recommendation was we needed to address assay performance, as Dr. Jeffrey Siegel just nicely pointed out. Evidentiary assessment matters. What they were saying is, as I've been noting here, we need the net effect of the intervention of the biomarker to predict the net effect on the clinically important endpoint. And context of use matters. A replacement endpoint cannot be readily extrapolated across other settings. For example, in coronary heart disease, LDL cholesterol is a validated surrogate endpoint for MI death, the clinical endpoint for statins. Now, if you have a new class of agents, that new class of agents may not have the same effect that statins had on other causal pathways, such as HDL cholesterol, or the magnitude, breadth, and duration of effect matters. Or maybe this new class has unintended effects of activating the renin-angiotensin system, increasing blood pressure. So in essence, then, why use replacement endpoints? Well, it's been clearly motivated that they can allow us to get answers in smaller sample sizes and shorter duration. We can get more timely results to give people a choice. And yet, timeliness cannot come at the expense of reliability, because the goal here isn't just to give people a choice, it's to give them an informed choice. And so in essence, then, what are some uses of biomarkers as replacement endpoints? And a biomarker only needs to be a patient level correlate to be useful for diagnosis and prognosis. It can also be obviously a measure of biologic activity in a proof of concept trial, or as supportive evidence in a registrational trial. The challenging issue is, when can we use them as replacement endpoints for registrational evaluation? And the situation matters. If it's simply refining the dose and schedule for an already established intervention, it could be rather straightforward to establish the utility of the biomarker. It could be for generalizing results to broader categories of patients, although we saw in the PAH setting, going from adults to children could be still problematic. So it's more challenging. It's even more challenging to justify replacement endpoints for new therapies, especially when these are in new classes. And so finally, then, the validation of biomarkers, what would be future steps? We need continued evaluation in aggregation of trials, meta-analysis of data across trials, where each of these trials should be reliably evaluating the efficacy of the OA therapies. And our meta-analysis should be independent of the level of trial positivity. It should be inclusive, where each trial should provide a reliable assessment of the intervention's effect on biomarker endpoints. So we need standardization of assays, and on direct measures of how people feel, function, and survive. And so we need to then increase the number of properly controlled studies of treatment, not only to increase the availability of proven, safe, and effective therapies, but to support the research efforts regarding validation of surrogates. So these should be ideally randomized, having standard of care controls, and evaluating effects on both the biomarkers and the clinical endpoints to understand the relationship between effects on biomarkers and effects on clinical endpoints. And this is particularly important when studying new classes of interventions. So in conclusion, a patient-level correlation is a necessary but not sufficient condition for a biomarker to be a validated surrogate. And that's also true when we're looking at validating a biomarker as a replacement endpoint for accelerated approval. A biomarker, to be reasonably likely to predict clinical benefit, does need to be a correlate of risk. But it's also important that it have substantial magnitude and duration of effect on that biomarker, and that the biological plausibility that that biomarker captures the treatment effect on the principal causal pathway is necessary. And there also needs to be likely evidence of an absence of influential unintended effects. So in simple terms, a correlate does not a surrogate make.